Now we have studied or various experimental evidences were there regarding the semi-conservative nature of the DNA replication. Before we start with the actual process or the mechanism of DNA replication, let us try and understand so as to have an idea what type of enzymes are actually involved in this particular process. There are chiefly 8 type of enzymes. Out of these 8 type, these 2 are important, but primarily if we say these 6 are the enzymes which are actively involved in the DNA replication. So those are classes of enzymes, that is helicases, topoisomerases, single strand stabilizing protein, that is SSBs, ligases, primases, DNA polymerases, that is 1, 2 and 3. So we have chiefly these 6 which are actively involved in the DNA replication process, because at some stage or the other of the replication process, these enzymes are taking part. Whereas the nucleases and the phosphorylases are important, but they are not actually involved in the process of replication. They are mainly involved, like phosphorylases, if we say, phosphorylases enzymes help in the addition of the phosphorus. Nucleases, they help in the proofreading, that is the DNA replication is or the process is perfect that way. So we'll talk about these 6 enzymes mainly which are involved in the process of DNA replication. Let us first start with helicases. Now you all know the DNA strands are coiled around each other, right? So these enzymes work on the hydrogen bonds. If the hydrogen bond between the two strands which are coiled with each other, if those hydrogen bonds break, as a result, what will happen? The opening up of the DNA fragment will take place. That means the two strands can then easily be separated. So the two strands open up with the help of helicases, and these helicase enzymes help in breaking up of the hydrogen bond. Then we have the topoisomerases. Topoisomerase is a class of enzyme that help in the release of tension. Now, when the strands are separated after the breaking up of the hydrogen bond, the strands are still in the coiled form, right? They will be still in the habit of coiling. So this topoisomerase enzyme prevents the recoiling, and it cuts, rotates and reseals the strands at the same point. Now this, as a result, since they are broken up into smaller fragments, these will not be coiled. So they help in disentangling of the DNA coils and the supercoils. Then we have the single strand stabilizing protein, that is the SSB. The SSB, now after what has happened, it is going stage wise. First the hydrogen bonds are broken up. As a result, the two strands are separated. Now, as the topoisomerase help in maintaining or avoiding the, we can say they help in avoiding the recoiling of the strands back. As a result, the two strands remain separate. But now again, these strands have a tendency, as I said, so SSBs give stability to the two strands. These proteins, they will bind to the single strand parts of DNA, and where the two strands are unbound, and they provide stability to the single strand and also prevent the recoiling. So this reduces the tension, and this prevents the recoiling by binding to the single strand DNA. Then we have the next class of enzyme, which is ligases. Ligases are very important because these are the enzymes which catalyze the joining of the short fragments of DNA by the formation of phosphodiester bonds. So when the bonds are broken, the fragments are there, these fragments are joined with the help of ligase enzyme, and this is always between the 3-OH group, 3-OH group and 5-phosphate group. You know it. So this ligase is an enzyme which helps in the joining. Next comes the primases. Primases actually, when we'll study, the DNA synthesis does not take place at one go. It happens in the formation of primers. And what are primers then? It's kind of a base they are getting ready. Primer is just the base. Before you start with the final wall painting, that is the painting of the wall, whitewash, first the primer has to be done. So it's kind of a preparation. So primers are actually the short stretches of RNA on which the nucleotides are added during the polymerization. So that is the formation of the new DNA will finally take place. So this class of enzyme help in the synthesis of primers. Next we have the DNA polymerase. DNA polymerases 1, 2 and 3. There are 3 classes of DNA polymerase, especially in prokaryotes. So we have 1 and 2 in the case they are involved in the repair mechanism. So we are not concerned mainly with the 1 and 2 because they are for the repair mechanism. DNA polymerase 3 is actively involved in the DNA replication process. It's a larger molecule as compared to 1 and 2, and also it helps in the synthesis of DNA. It works as a dimer. Dimer means it can replicate both strands simultaneously. That means it can help in the multiplication of both the strands together. So this DNA polymerase is of our interest, which is DNA polymerase 3. So we have these different classes of enzymes, and these enzymes are actively participating in the actual process of DNA replication.